Hi gang! So today's topic is gene regulation. Do you remember back when we talked about differentiation? So embryos, that's this little tiny group of cells here that's going to form a baby, they start off with stem cells that do not yet have a specific job. The stem cells differentiate throughout development, becoming specialized cell types with specific jobs and characteristics. So once again, every cell in your body has the same DNA. So as stem cells grow and they divide by mitosis, the daughter cells get identical DNA. Differentiation occurs when different sets of genes within that DNA are expressed or turned on. When a gene is expressed, that means that RNA polymerase here can access the gene's promoter and begin transcription, resulting in an mRNA copy of the gene, which then allows a protein to be produced. So it's really all about whether or not this RNA polymerase can get in there and access that gene to make a copy in mRNA. So in eukaryotes, controlling which proteins get made in a cell usually happens by controlling transcription. There are other ways, and it's a little bit different in prokaryotes. But in us, there are these DNA binding proteins known as transcription factors. So that's just a protein that'll come in and interact with or bind to DNA. And it regulates a gene's expression by controlling RNA polymerase's access to that promoter. So there's a couple different kinds. Some transcription factors determine how tightly or loosely packaged the DNA is. So controlling physical access to the gene. So for example, here is a gene that's nice and open. RNA polymerase could get in there and transcribe that. If it gets kind of bunched up and coiled up a little bit more, it's going to be harder for RNA polymerase to get in there, so maybe not very much of the protein will get made. And then it can also get like super wound up, and RNA polymerase can't get in there at all to make that mRNA, to make the protein. Some other transcription factors help to basically attract RNA polymerase to the promoter by binding to regions called enhancers. So remember, the promoter is that spot where RNA polymerase is going to bind and start transcribing the gene. But it's not very good at finding it by itself, so usually multiple transcription factors have to bind to the enhancers, which are other areas near the gene where proteins, transcription factors can bind and help RNA polymerase get started. And then there's other transcription factors known as repressors that can keep certain genes off by binding to the DNA and either blocking the promoter or blocking the enhancers. So stopping this RNA polymerase from going on and transcribing the gene. So different types of cells have different transcription factors at work. So different transcription factors means different proteins are going to get made, which means the cells are going to have different characteristics and abilities. So for instance, here's a stem cell. It's going to do some mitosis and make a copy of itself. And maybe this cell will get some neuron genes turned on and other genes turned off. So it will become a neuron. So it's got special proteins inside that help it to send messages and make it the cool shape that it is. And then this other cell is going to maybe experience some different signals. And so it's going to have epithelial or skin cell specific genes turned on. So these genes are going to help it make it nice and flat and connect tightly to the other skin cells and make our skin waterproof and all the good things that our skin cells can do. So hopefully now that you understand a little bit more about DNA and how it gets turned into mRNA, and how that mRNA gets used to make proteins, maybe it makes a little more sense now that these cells can undergo differentiation. And so turning on genes just means that mRNA can get made and protein can get made, and then the genes that get turned off, well, those proteins are out of luck. So that's it for today.